Hi, welcome back to Biochem Talk. In this video, we discuss about gluconeogenesis. Gluconeogenesis is the synthesis of glucose. It helps to maintain blood glucose concentration during fasting and starvation. Gluconeogenesis takes place mainly in liver, kidney, and intestine. Gluconeogenesis occurs in microorganisms, fungi, plants, higher animals, and human beings. Cells such as RBC, neurons, renal medulla, and islands depends upon gluconeogenesis for their activity. Gluconeogenesis synthesizes glucose from non-carbohydrate sources such as pyruvate, lactate, propionyl-CoA, glucogenic amino acids, and glycerol. Pyruvate and the lactate are the end product of glycolysis. Out of 10 enzymes in glycolysis, 3 enzymes are reversible. They are pyruvate kinase, phosphofructokinase, and isokinase. Pyruvate kinase converts phosphoenol pyruvate to pyruvate, but it cannot convert pyruvate to phosphoenol pyruvate. So, pyruvate is converted to phosphoenol pyruvate by 3 enzymes such as pyruvate carboxylase, malate dehydrogenase, and phosphoenol pyruvate carboxykinase. First enzyme, pyruvate carboxylase, present in the mitochondria. So, pyruvate enters into mitochondria by mitochondrial transporters. In mitochondria, pyruvate is carboxylated to oxaloacetate by pyruvate carboxylase. This reaction utilizes one molecule of ATP. As there is no mitochondrial transporters for oxaloacetate cannot travel across mitochondria. So, oxaloacetate is converted to malate by malate dehydrogenase using NADH coenzyme. Malate can travel across mitochondrial membrane by malate aspartate shuttle. Malate enters into cytoplasm. Malate is again converted to oxaloacetate by malate dehydrogenase using NAD as coenzyme. In the cytosol, oxaloacetate is converted to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase enzyme. Now let's see gluconeogenesis from lactate. Glucose is converted to lactate during anaerobic glycolysis. RBC cells synthesize lactate during insufficient oxygen, while skeletal muscles synthesize lactate during strenuous exercise. Lactate is converted to phosphoenol pyruvate by 3 enzymes. They are cytosolic pyruvate dehydrogenase, pyruvate carboxylase, and mitochondrial phosphoenol pyruvate carboxykinase. In the cytosol, lactate is converted to pyruvate by pyruvate dehydrogenase using NAD as coenzyme. Pyruvate is transported into mitochondria by mitochondrial transporters. Pyruvate is converted to oxaloacetate by pyruvate carboxylase as explained before. Oxaloacetate is converted to phosphoenol pyruvate by mitochondrial phosphoenol pyruvate carboxykinase enzyme. Phosphoenol pyruvate is then enters into cytosol. Phosphoenol pyruvate is converted to fructose 1,6 bisphosphate by reversible enzyme present in glycolysis. Fructose 1,6 bisphosphate is converted to fructose 6 phosphate by fructose 1,6 bisphosphatase enzyme. Fructose 6 phosphate is converted to glucose 6 phosphate by reversible enzyme in glycolysis. For a detail, please see glycolysis video which I have updated before. Glucose phosphate is converted to glucose by glucose phosphatase enzyme. Next we see synthesis of glucose from propanol CoA. Sources of propanoate are valine, isoleucine, leucine and cellulose. Beta oxidation of odd-chain fatty acid gives propanol CoA. Propionate gives propanol CoA by acyl CoA synthetase enzyme using ATP as coenzyme, magnesium as cofactor. Propanol CoA is converted to D-methylmalonyl CoA by propanol CoA carboxylase using biotin and ATP as coenzyme. D-methylmalonyl CoA is converted to L-methylmalonyl CoA by methylmalonyl CoA racemase. L-methylmalonyl CoA is converted to succinyl CoA by methylmalonyl CoA mutase using vitamin B12 as coenzyme. Succinyl CoA is one of the intermediates of citric acid cycle. Succinyl CoA is converted to oxaloacetate by reversible enzymes present in Krebs cycle. As explained before, oxaloacetate is converted to propionyl pyruvate and then to glucose. Next we see synthesis of glucose from glucogenic amino acids. Glucogenic amino acids such as alanine, serine, glycine, cysteine, threonine, and tryptophan is converted to pyruvate. Other glucogenic amino acids are converted to alpha-ketoglutarate, succinyl CoA, fumarate, and oxaloacetate, which are the intermediates of TCA cycle. They are converted to glucose as described before. Next, we see the gluconeogenesis from glycerol. Glycerol is formed from fat lipolysis in white adipose tissue. Glycerol kinase enzyme is present in the liver. So, glycerol enters into liver. Glycerol kinase enzyme converts glycerol to glycerol 3-phosphate. Glycerol 3-phosphate is converted to dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase. Glycerol dihydroxyacetone phosphate is converted to glucose as explained before. Thank you. If you like, please subscribe to Biochem Talk.